Good afternoon, everyone. So welcome to the Nobel Prize Popular Science Lectures presented by the School of Science and kindly hosted by the IAS. My name is Kenward VONG, and I'm currently an Assistant Professor of Chemistry here at HKUST. And I'm here today to talk to you about the works of the recent recipients of the Nobel Prize in Chemistry. So to be honest, I'm probably not the most qualified person to be giving this lecture. Ideally, we would be having the Nobel Prize laureates themselves, followed by more recognized names in the field. Having said that, I think it really speaks to the utility of this chemistry that even someone like me has done the click reaction before. So not only very important to the field of bioorthogonal chemistry, but the click reaction has become very important to many disciplines across many disciplines. So without further delay, I'd like to get started and talk to you about click chemistry and bioorthogonal chemistry. So as you may know, this year's Nobel Prize in Chemistry went to Professors Morten Meldal at the University of Copenhagen, Professors Barry Sharpless at Scripps Research, and Professors Carolyn Bertuzzi at Stanford University. Now, Professor Meldal, currently age 68, did the bulk of his Nobel Prize-winning work at the Carlsberg Laboratories in Copenhagen. Within the international peptide community, he's a really well-respected name because his principal interests fall in the area of synthetic peptide methodologies. In fact, in the seminal paper where he described the copper-click reaction, he was trying to sell it as a way to do solid-phase combinatorial peptide synthesis. Next, we have Professor Barry Sharpless, I think who needs no introduction. This is actually his second Nobel Prize. He won his first Nobel Prize at the age of 60 in 2001, and this was to recognize his work with chirally catalyzed oxidation reactions. So this reaction, such as the Sharpless epoxidation, Sharpless asymmetric dihydroxylation, among others. So he actually won the Nobel Prize in 2001, but only 1 year later, in 2002, he would go on to publish the seminal paper that would win him his second Nobel Prize. So that's coming now at the age of 81. And finally, we have Professor Carolyn Bertuzzi, who's currently age 56. To put it lightly, she's a powerhouse in bioorthogonal chemistry. I think first and foremost, she's a glycobiologist who really needed to develop her own chemistry to study the systems that she wanted to. So as a little background, cell glycosylation is a very difficult thing to observe because of its dynamic and diverse nature. So Professor Bertuzzi basically had to come up with the modified Staudinger ligation and the strain-promoted click reaction in order to study these systems. So as you can see at the top, the Nobel Prize Committee basically awarded these three individuals for the development of click chemistry and bioorthogonal chemistry. So today's lecture, I'll be talking on these subjects. So I'll be breaking my talk into three different segments. First, I'll be talking about copper click chemistry, basically who are the major players involved. Then I'll be talking about the applications of this copper click chemistry to biological systems. So essentially the field of bioorthogonal chemistry, followed by just a very quick talk about the future directions of this field. Okay, so before we can talk about click chemistry, I think it's very important we recognize the vital contributions of Professor Rolf Wiesken. So as a little background, Professor Wiesken's a German chemist whose early career coincided with the Second World War. So I remember reading that after the lab of his PhD supervisor was destroyed in the air raids, he had to move to a little town 50 kilometers south of Munich, and he basically made his own makeshift laboratory to finish his experiments. So that sort of shows his dedication to his craft. After the war, he then moved back. Eventually, he moved back to the University of Munich, where he helped with the modernization of the laboratories there. So it's there that he did his best work. So he published over 600 papers that range from 1,3 dipolar cycloadditions, 2+2 cycloadditions, and electrocyclic reactions, among many, many others. So of course, today we're here to be talking about the azide-alkyne Wiesken cycloaddition. So it's a really simple reaction. It basically has two components. It has a 1,3 dipole, which comes as a form of an azide, and it has a dipolarophile, which comes in the form of this alkyne. So when you put these two together, what happens is a 3+2 cycloaddition. So basically, this electrical charge is going to attack this pi system, and then the pi electrons are going to form this carbon-nitrogen bond while neutralizing this positive charge. As a result, what you get is a 1,2,3 triazole. Now, this reaction is not perfect. It has some flaws. I think one of the major flaws is that it's not regioselective. So what you're going to get is actually a mixture of the 1,4 and 1,5 adducts. And this is solely dependent on the position of this sort of yellow circle during the cycloaddition. Another major flaw with this reaction is that it's really slow. So you basically have to cook this thing under reflux overnight in order to get it to work. But in terms of the chemistry, this was definitely interesting because at that time, which is, it says here 1963, access to these 1,2,3 triazoles was still quite rare. So from a chemistry perspective, it was definitely interesting. But you can probably understand why in terms of when you look at its potential adaptability into biological systems, that it's probably not ideal. Fast forward to the year 2002. This is the year that the groups of Meldal and Sharpless independently published their copper-1 catalyzed azide alkyne cycloaddition, or CuAAC. So this reaction is very similar to the Huiskin cycloaddition. The only major, it has the azide and the alkyne, combining it to make this 1,2,3 triazole. The only major difference is that they're using now a copper-1 catalyst. Okay, and so by adding this copper-1 catalyst into the mixture, there are three major changes. First, this reaction's a lot faster. It's 10 to the power of 7 times faster. So instead of doing it overnight, this can be done within less than an hour. Second, this reaction can be now done under room temperature, so no longer does it need to be heated. And lastly, this reaction is now very regioselective. It's only going to form this 1,4 adduct. Now, before I move on to the next slide, I should also mention that the catalytic component of this reaction is the copper-1 species. However, copper-1 under aqueous conditions is not stable. So in practice, usually researchers, they add a copper-2 species, like copper sulfate, along with a reducing agent in order to generate the copper-1 in situ. And in this manner, they can get by the sort of the instability of the copper-1 in water. Now, for the chemists in the room, probably the most important thing that you probably want to hear is sort of what is the mechanism. So there appears to be two mechanistic pathways. The first pathway was proposed by Sharpless and his colleagues. And what they sort of based this on was that it was acting through a mono-copper complex. Now, more recently, through kinetic experiments and sort of DFT calculations, it's been proposed that this is likely going through a bis-copper complex. But in general, I think the arrow pushing stays very similar. So what happens is that the alkyne interacts with this copper. It forms this pi complex. And in forming the pi complex, the pKa of this terminal proton drops more than 10-fold. So the pKa becomes 9.8. This makes it more abstractable, so your copper-1 acetylide can now be formed. Now this copper-1 acetylide can now interact with the azide, and it does it through this alpha nitrogen here to give us this sort of intermediate. Now the next step is ring formation, so make this copper-3 metallocycle, which undergoes reductive elimination to give us this triazole copper derivative. Now, obviously, the final step is then just protolysis, where the acid frees up the copper-1, giving us our 1,4 triazole. Now, I should definitely mention that although copper is very specific for the 1,4 triazole, it is possible to make the 1,5 triazole. And to do this, we just have to use the beautiful ruthenium catalysts that have been developed by none other than our own Professor Jia in the chemistry department. I'm sorry, due to time constraints, I had to cut out a slide on the ruthenium click, but I'm sure most of you are already very familiar with his work. So with the development of the copper-1 catalyzed azide alkyne cycloaddition, this was basically the birth of click chemistry. So for those of you who are not familiar, click chemistry is a term set out by Barry Sharpless to describe reactions that join molecules together like modular building blocks, sort of like clicking together like Legos. So in order to meet the requirements to be a click reaction, Barry Sharpless basically outlined that it needed to be modular in nature, wide in scope, high yielding, produces inoffensive byproducts, and proceeds under friendly reaction conditions. And by friendly, what I mean is basically open to air, in the presence of water, and also at room temperature. So with the copper-catalyzed azide alkyne cycloaddition, this was basically the first reaction to meet all of these requirements, so it became a pioneering reaction for click chemistry. Now I should also mention that after Meldal and Sharpless's work, numerous groups also went ahead and developed their own reactions that meet these requirements. So now we actually have a very small library of reactions that we can call click reactions. Now I'll very quickly touch on these reactions at the end of my talk, because for this particular lecture, I want to focus on copper click chemistry. Now we all know what click chemistry is, right? So some of you may be wondering, what the heck is bioorthogonal chemistry? It's kind of a weird word, don't you think? It's got these two O's here. And if you only write it with one O, then it becomes a completely different thing. It becomes a mathematical term. And so sometimes in literature, you may see these two O's separated by a hyphen. But how it's intended to be written is shown here. And this term was actually coined by Carolyn Bertuzzi, although its first appearance in literature came in this 2003 PNAS paper. Bertuzzi states that she's been using this term since the late 90s in various public presentations. So before we can describe bioorthogonality, I think it's very important to understand what orthogonality means for a chemist. So in chemistry, orthogonality basically describes a set of reactions or individual reactions that can be performed independently in the same pot. So for example, the orange reactants come together to form this orange product, and these green reactants come together to form this green product. But if you take an orange reactant and a green reactant and you try to react them together, nothing will happen. And that's basically orthogonality. Basically, these reactions are specific enough that they will not interfere or be interfered by other reactions. So now, when we're talking about bioorthogonality, it's sort of a similar concept. You have a chemical reaction that can occur inside of living systems that cannot interfere or be interfered by these native biological processes. So what does that mean? So for example, what are some of the functional groups you see in a biological system? You see amines, you see hydroxyl groups, you see thiols. Those are all functional groups that are found on the surface of proteins and also metabolites. So if a reaction is going to be bioorthogonal, it definitely cannot react with the amines, hydroxyls, and thiols. Another test would be water, right? Our bodies are made of 50 to 70% of water. So for a bioorthogonal reaction, it should definitely be able to proceed in water, preferably at a pH of 7.4. So shown here on the slide is the very first example of the copper click reaction being performed with a biological system. So to explain this slide, Professor M.G. Finn had previously worked with these cowpea mosaic viruses. They're really interesting. These are sort of a plant virus where the outer shell is composed of 60 copies of 2 protein subunits. Now, each of these subunits only have one lysine that will basically protrude into the extracellular environment. So as a result, when this sort of virus particle forms, you're going to have 60 lysines on the surface. So what Professor Sharpless did was then he coupled compound number 1 to the surface of the virus particle, and so he could get basically an azide-decorated virus particle. He then incubated with this alkyne-containing dye. Of course, the structure of the dye is shown here under these conditions. Now, you'll notice that for these conditions, they're using a copper-2 species along with the reducing agent so that they can basically generate the copper-1 in situ. Now, as a result of this work, they were able to show that the copper click chemistry could indeed label the dye 60 copies of the dye on the surface of this virus. So they basically got quantitative yield. So as a result of this work, basically it proved to the scientific community that the copper-click reaction could indeed truly be compatible with biological systems. Now, with all good things in life, there's always a catch, right? And the major disadvantage of copper-click chemistry is unfortunately the copper itself. Free copper-1 species are very toxic because they can cause a lot of oxidative damage, and it does this through a Fenton-like reaction. So basically, the copper-1 can get oxidized to a copper-2 via this hydrogen peroxide. Now, where does that extra electron go? It basically gets inserted in between this peroxy bond, which will break apart and form this hydroxyl radical with this one unpaired electron, and with this hydroxy anion with this pair of electron here. Now, the hydroxy anion is pretty harmless, but the problem is the hydroxyl radical. These are very, very reactive species. So it's basically going to indiscriminately react with any biomolecules in the system. So if the concentration of this hydroxyl radical is high enough, it's going to cause a lot of damage to the cell. So a very good example of copper toxicity is with Wilson's disease. So this is an inherited disorder that causes copper accumulation to the liver, brain, and other vital organs. In fact, you can see on this picture on the right, this is the eye of someone with Wilson's disease. And you can see there's a brown ring, right? This is what's known as a sunflower cataract, and it's just basically copper deposits, accumulating in your eye. It's kind of freaky, right? But besides these aesthetic changes, obviously having excess copper in your liver and brain is going to cause a lot of damage. Okay, so we all now know that copper is bad, right? So how do researchers deal with this? How are researchers going to carry out their experiments without poisoning the very biological system they're trying to analyze? Well, there are basically two approaches they can use. The first approach is to create a metal complex that can stabilize the copper-1 oxidation state. The reason for this is that if the catalytic copper-1 oxidation state is, catalytic copper-1 is stable, it's much more available to react and for the cycloaddition. So as a result, researchers can then use much smaller amounts so that they can get under toxic thresholds so it won't kill the biological system. Another strategy is just to get rid of the metal altogether, right? No metal, no problem. So from here, I'll just be talking about 3 adjustments that researchers have made in order to make the copper-click chemistry less toxic and more compatible with biological systems. So I'll first talk about ligand-assisted copper-click chemistry. Basically, this was developed by Barry Sharpless and his colleague Valerie Fokin. This is basically in their preliminary studies for the copper-click chemistry. What they found was that certain polyvalent substrates were reacting unusually high. It was almost like they were autocatalytic. So what they did was they designed a series of experiments to test a range of ligands supplemented into the reaction to see their effects. And what they found was that triazoles themselves, triazoles which are the very product that this click reaction is making, the triazoles themselves was accelerating the reaction. So you can see here with this reaction between the alkyne and the azide, again, just to point out that they're using a copper-2 along with the reducing agent to make the copper-1 in situ. But when they have no ligand here, the yields are only about 21%. However, when they add their triazole and only 10 molar %, yields shoot up to 99%. So along in their studies, Sharpless and Fokin basically came to this ligand, which they dubbed TBTA. And what this basically is a polytriazole compound that's very good at stabilizing the copper-1 oxidation state. So here, let's look at the experiment. Basically, again, they're putting the alkyne with the azide together. But in this case, they're directly putting in a copper-1 catalysis. They're not putting in copper-2 or any sort of reducing agent. Now, as I mentioned before, copper-1 is very unstable in water. What's going to happen is it's going to undergo disproportionation to form this copper-0 and the copper-2. So it makes sense that without any sort of reducing agent or any stabilizing ligand, that if they don't add a ligand, their yields is basically 1%. However, when they add their TBTA, this ligand can basically recover the activity and give you about 84%. And what's happening is it's stabilizing the copper and striking a fine balance between reactivity and stability. So following up on these observations by Sharpless, there was an opportunity to do what chemists do best, and that's to make derivatives, right? So for example, Peng Wu, who's now at Scripps Research Institute, made a series of accelerating ligands to see if he could improve reactivity under physiological conditions. And he reasoned that he can improve reactivity by making these ligands more water-soluble. So that's how he got to these compounds, THPTA, which is functionalized with an alcohol here, BTTES, which is functionalized with this sulfonic acid, and BTTAA, which is functionalized with this carboxylic acid. Now, as you can see in the reactivity chart in the middle, here they're just comparing the rates of these different ligands, and it's very clear that the BTTAA and the BTTES are far superior in reactivity compared to the BTTA, which is the ligand I just showed in the previous slide. So encouraged by these results, the Wu group then tried to prove their utility in a biological system, so they basically labeled the surface of zebrafish embryos. So the images here on the top are fluorescent images, and the ones at the bottom are just bright field images. So as you can see, it's hard because there's no color, unfortunately, but you can see that the surface of their embryos were all labeled using click chemistry. Okay, so another accelerating methodology that was discovered is the chelation-assisted copper click chemistry. So this is a principle that was established by Professor Lei Xu at the Florida State University. Basically, what he found was that if reaction rates could be improved if he used an azide that was tethered to a nitrogen-containing auxiliary, okay, so such as this pyridine moiety. So basically what happens is that by coordinating with having this auxiliary, it will coordinate with the copper, and that in turn will speed up the bond formation between the copper and the azide. In addition, it said in that paper that the electrophilicity of this azide is going to be enhanced, and therefore it lowers the activation barrier for cycloaddition. So let's look at some of the experimental results. So here they're coupling this alkyne with two different types of azides. Obviously, when they have the azide with only a phenyl group, this has no coordination, right? It has no nitrogen. So obviously, the yields are low. Now, when they're using this pyridine instead that has the nitrogen to coordinate, yields can therefore be enhanced and recovered. Okay, so to prove the biological utility of the chelation-assisted copper click chemistry, Professor Alice Ting at Stanford University basically used it to label the cell surface proteins. So in this methodology, she basically had her protein of interest express this 13 residue LAP sequence. Now, this LAP sequence is a substrate for the prime ligase. So what's going to happen is that compound number 3 can be selectively coupled to the end of this LAP residue. And so you will have your protein of interest expressing this picolyl azide moiety. Next, by putting in this the alkyne-containing dye along with the copper, you can then selectively click to give your dye to be labeled on the protein of interest. So using this technology, Professor Ting basically labeled human embryonic kidney cells. And so you can take a look at the data here on the right. So let's look on the far right. So what's shown here are cells that express the protein of interest. So only the ones that are fluorescent green have the protein of interest. So that means only those cells will have the azide. All these other cells will not have an azide, right? So when they take these cells and incubate it with their alkyne-containing dye, what they find is that only those cells that are fluorescent green will have labeling on the membrane surface, and that's what's shown by these purple rings. Okay, so that's kind of cool. Another thing that Professor Ting found was that by using the picolyl azide, they could decrease the concentration of the copper that they needed to get these results. So let's say if they were just using a regular azide, they would need 100 micromolar of copper to get these results. However, if they use the picolyl azide, the copper can be reduced by more than tenfold, so they only need about 10 micromolar of copper to get these results. So that sort of just proves how using the sort of the picolyl azide along with the sort of the copper-1 complex can help to decrease the amount of copper needed to do these experiments. Finally, I want to move on to the last methodology to deal with the copper toxicity, and that's to just get rid of it altogether. And so shortly after the works of Meldal and Sharpless was published, Professor Bertuzzi had a hypothesis. She reasoned that perhaps a strained alkyne within a ring system could activate the alkyne enough that it could undergo the cycloaddition without the need for copper. So this is how the strain-promoted 3+2 azide alkyne cycloaddition, or SPAAC, was conceived. Now, through their experiments, they found that the cyclooctane, which has 8 carbons in this ring, was the best. If they used rings that were smaller, they were too unstable, and if they were using rings that were larger, they were just not reactive enough. Now, one of the problems with SPAAC is that it's much less reactive compared to the copper catalyzed reaction. So you can see here for the cyclooctane, the second order rate constant is only about 0.0012. Now, for comparison, the copper click chemistry can be done, its second order rate constant can go up to almost 100. So you can see that there's a huge trade-off in removing the copper. So what chemists do best is Professor Bertuzzi basically set out to try to improve these strained alkynes. And one method that found a lot of success for her was through fluorination. So basically, she made this difluorinated molecule known as difo, and by doing so, she could improve reactivity by 43-fold. Now, the reason for this is because these fluoro substituents are acting as electron withdrawing groups. So coupled with the ring strain, it makes it much more reactive. Another modification that she looked at was making these dibenzocyclooctanes. So here she's basically putting these 2 benzo groups on each side of the alkyne. And the reason for doing this is basically to increase the strain energy. Because now you're putting 4 sp2 carbons within the ring, so it's going to be much more strained, and rigid and strained. And as a result, she could, for compound, the compound dibo, the rate of reaction could be increased also by 42-fold. Now, when Bertuzzi tried to put another aromatic ring on this, the cyclooctane, what she found was that these compounds were very, very unstable. So what she set out next to do was to find other modifications that had varying degrees of double bond character. And that's how she came across Barak. Basically, amide bonds have significant resonance structure. So by putting this amide bond within the ring, basically she found it could have a good balance between reactivity and stability. And as a result, Barak has a sort of over 800-fold increase in reactivity compared to the cyclooctane. Now, although its second order rate constant is still only roughly 1, this is actually more than enough to carry out experiments for biological studies. Okay, so as a result of the fine work of the researchers I just mentioned, there are now several biocompatible methods to do copper click chemistry. So now I just want to move on and talk about sort of the applications of copper click chemistry to the field of bioorthogonal chemistry. So there are 3 main biological applications I'll talk about today. One will be metabolic labeling on the whole cell scale. The other 2 will be protein bioconjugation and activity-based insertion on the single protein scale. So let's get started with metabolic labeling. So before I talk about metabolic labeling, I think I should introduce what metabolic incorporation is. Has anyone ever heard the term, you are what you eat? Well, this is basically, metabolic incorporation is basically that. This is a phenomenon discovered by researchers where they can basically incubate these unnatural biomolecules to the cell, and the cell will naturally uptake these molecules, modify them, and then incorporate them into the biosynthetic pathways that produce glycoproteins, lipids, and DNA. So for example, let's look at this glycan. Basically, you can make this acetyl-protected glycan that has either an azide or an alkyne, as represented by this blue shape here. And when you feed this into the cell, it'll get uptaken, and of course, the acetyl groups will get deprotected. It's going to get modified and activated with a UDP moiety so that it becomes basically glycan building blocks. So since glycosyltransferase is a very promiscuous enzyme, it can then accept these as substrates and basically make glycoproteins where you have azides and alkyne handles on the surface. The same principle also applies for lipids and DNA. But due to time constraints, I won't be talking about these. I'll just be focusing on glycans. Okay, so metabolic glycan labeling is basically a technique that has been pioneered by Carolyn Bertuzzi. Her group has done a lot of work within this field. So what's shown here is actually one of many, many examples. And now I'm highlighting this particular example because I think it's a really great experiment showing how this amazing chemistry could be used to tell us something new about biology. So in this experiment, the zebrafish embryos were basically injected with this azide-containing N-acetylgalactosamine. Basically, these would get uptaken by the cells and then modified and rearranged and then expressed on the surface as glycoproteins. So basically, in the zebrafish, the surface of their cells will have this azide. Now, the really cool thing about this experiment was that from the 3rd to 60th hour, these zebrafish embryos were injected with the azide. Then, from the 60th hour onwards, they stopped. And from the 60th to 61st hour, they only injected the strained alkyne with a fluorophore that corresponds to the orange in these images. And now, after that 1 hour, they then stopped injecting these particular probes, and from the 61st to 62nd hour, they only injected strained alkynes with a fluorophore that corresponds to the green color in these images. So the point of these steps was basically to observe the arrangement and pattern of new glycan formation during development. So as you can see in these images, from the 61st hour onwards, within the mouth region, pectoral fins, and the olfactory organs, you can see a clear, you can clearly see that new glycan distribution is sort of localized to specific regions, and it's sort of done in a non-uniform manner. So unfortunately, scientists still don't know what this means, or they've only uncovered that this is what happens. But this is definitely something that would not have been possible to uncover without the power of click chemistry. I don't know of any other technique that can sort of analyze glycan development in this manner. Okay, so another example by the Bertuzzi lab is shown on this slide. In this case, she increased the difficulty by doing glycan metabolic labeling in mice. So for this experiment, she basically used these azide-containing mannose-amines, and she just injected them into the mice. Now, as a little background, mannose-amines are basically precursors to sialic acid. Sialic acid is a sugar found in many different types of cells, but as a quick primer, it's abundantly found as terminal sugars in the mucus glycans in the intestines. So basically, after she injected the azide, she then injected this flag tag cyclooctane, the strained alkene. And then, obviously, the flag tag would then be clicked to the surface of organs that are rich in sialic acid. Now, therefore, it makes sense because the intestine is rich in sialic acids, so when they basically sacrificed the mice and then analyzed the organs, they could see that it was mainly, the intestines had a very, very heavily tagged in this case. Okay, so although there's a lot more I can talk to you about with regards to metabolic labeling, I just want to move on to the next topic, which deals with click chemistry that's done more selectively on the single protein level. So first, before I talk about an example of click chemistry being used for protein bioconjugation, I first want to introduce the concept of antibody drug conjugates. So for those of you who are not familiar, antibodies are proteins produced by our immune system to recognize and bind foreign molecules. Now, since certain biomarkers are overexpressed on cancer cells, researchers have very elegantly designed therapeutic antibodies that will recognize these biomarkers. And in doing so, they can then elicit specific cancer cell death. Now, to take things a step further, we can make antibody drug conjugates. And as the name implies, we're basically functionalizing these antibodies with a drug via this linker. Now, a particular challenge in the development of antibody drug conjugates relates to the method of conjugation. So typically, you only want to be conjugating your drug to the Fc domain. And traditionally, this is done by having sort of a lysine residue or cysteine residue that exists in this Fc domain, which you can then use to couple with your linker and drug. Now, unfortunately, this method is wholly dependent on these lysines or cysteines not occurring in the antigen binding domain. Otherwise, they would get in the way of recognizing the antigen. So as you can probably guess, this is where click chemistry and genetic code expansion can come into play. Basically, these antibodies can be produced to have a site-specific azide functionality. So if you site-specifically make an azide here in Fc region, you can then couple your alkyne-containing linker to the drug there, and so that only that region will have the drug. So you can basically control only maybe 1 or 2 drug molecules to be on the antibody. So I think that this case shown on the slide highlights the increasing importance of click chemistry. Although its adaptation is a little slow, antibody drug conjugates are now beginning to use more click-based conjugation methods. So currently, there are actually 2 such antibody drug conjugates under clinical trials. And I'll be talking about one of them. So STRO-001 is an antibody drug conjugate that targets hematological cancers through binding the CD74 receptor. And how they do it is basically at the 404 position of this antibody, they've incorporated this azide methylphenylalanine. This residue here highlighted in green. Now, following the click reaction with the cyclooctane, this cyclooctane will be linked with a PEG linker, which will also be attached to your drug moiety. In this case, it's maytansinoid. So by treating cancer cells with this antibody drug conjugate, they'll basically get internalized, and eventually this drug will be cleaved, and it can then exert its anti-tumor effects. So I know currently the number of cases that have made it into clinical trials is still very, very small. I mean, there's only 2 cases. I think it's only a matter of time before more protein conjugates that use click chemistry will be created, tested, and approved by the FDA. Okay, so for the final example of the bioorthogonal applications of copper click chemistry, I just want to talk to you about activity-based insertion. Before I get started, I want to pose this question. How does one identify if a protein binds a specific molecule? Now, if you're dealing with a scenario with a purified protein, it's actually very easy, right? You just incubate the purified protein with the molecule, and then hopefully you'll have a binding assay handy. So if there's an appropriate signal, you know that there's a binding event between the molecule and the protein. Now, since this is a purified protein, you should know the identity of that protein. Now, what if you have a scenario where it's a complex mixture of proteins? You can still incubate your molecule with this mixture, and you can still do a binding assay, and you'll still get a binding signal. But it's very difficult in this case to find out the identity of what's binding your protein. I mean, it could be just one protein binding your molecule, or it could be several different proteins binding your molecule. So how do scientists deal with this? So this is a technique that was developed known as activity-based protein profiling. Basically, it's a technique used to identify protein targets of small molecules in really complex mixtures, such as living cells. So how this works is that you first have a AB-PP probe here, and it has 3 parts to it. The first part is a recognition group. So this group basically directs the probe towards whatever protein you're trying to see what it binds to. So for example, if you're trying to find out a proteins that bind a specific peptide residue, you would put that peptide sequence here. Next, you would need a reactive electrophile. Basically, what the purpose of this reactive electrophile is that once the probe binds to its target protein, this warhead is available so that nearby nucleophilic residues can attack this and form a covalent bond. And in forming a covalent bond, basically the probe is forever linked to that protein. And finally, you have a reporter tag. And so the purpose of the reporter tag is to allow researchers to extract and identify what exactly is covalently bound to this tag. So in these experiments, basically, you have your sort of proteome. You would incubate your probe with it, and you give it some time. And of course, the probe would go and basically bind to whatever protein that it has high affinity for. And then, obviously, some sort of nucleophilic residue that's in the neighboring vicinity is going to form a covalent bond with the electrophilic warhead, which would cause a covalent bond to form. And now you can basically analyze the mixture. So if your reporter tag is fluorescent, great. I mean, you just have to run it on a gel. And so basically labeled proteins will be fluorescent, and you can identify them that way. If your reporter tag is a pull-down tag, basically like biotin, you can then extract them out and do MS-MS analysis and peptide fingerprinting to sort of determine the identity. Now, as great as activity-based protein profiling sounds, there is a flaw, and this issue stems from the reporter tag. So because the reporter tag is usually a fluorophore or it's a biotin, which is used for the pull-down assay, these are often very bulky, and they may hinder protein binding. So if your substrate channel is actually, if it's narrow, that reporter tag may actually sterically clash with the protein. And in doing so, researchers would miss out on identifying these proteins as protein targets. So in order to fix this problem, basically what you can do is you can replace the reporter tag with either azide or alkyne, because these are very small functional groups. They're not, they're not theorized to interfere with any binding. So once these new probes bind to your protein target, the nucleophilic residue can then form a covalent bond. And then after a covalent bond is established, you can then just incubate your mixture with an alkyne or azide reporter tag for however long is necessary to make this click reaction, and then you can analyze. So a very good example of this click assisted activity-based protein profiling is shown on this slide. In this slide, the CBER group wanted to find out the mechanism of duocarmycins. So as a little background, duocarmycins are anti-cancer drugs that were originally thought to act by binding DNA, and when it bound DNA, it would form a covalent bond and eventually cause apoptosis. Now, that theory was brought into question when eventually a duocarmycin derivative was made that lacked any DNA binding moiety. And that derivative actually had more activity than the parent drug. So basically, researchers thought that this drug was probably doing something else. So the CBER group basically used activity-based protein profiling to find out a mechanism for duocarmycins. So they synthesized this probe. Basically, on the left-hand side is the duocarmycin, and the cyclopropyl group is the reactive moiety. And of course, it has this alkyne, which is used for clicking later. So they incubated this probe with these lung cancer cells. And of course, they incubated for some time so that a covalent bond could be established. And then they lysed the cells. And from this mixture, they treated it with this azide containing rhodamine, along with copper. Now, after they ran the analysis, which in this case was a fluorescent STS page gel, they found one very, very strong fluorescent band here around 60 kilodaltons. And that turned out to be aldehyde dehydrogenase 1. And previously, that was an unknown binder for duocarmycins. And as a little background, aldehyde dehydrogenase 1 is a enzyme that's implicated in the stemness for cancer cells. So there are groups out there trying to develop drugs that will specifically target this enzyme. Okay, so since it seems like we're almost running out of time, I want to quickly wrap up and talk to you about where this field may be headed in the future. So as I mentioned before, there are many types of reactions that fit the description of a click reaction set up by Sharpless. So these include the modified Staudinger ligation, the oxim ligation, isonitrile click, and the tetrazine ligation. So although these are all wonderful tools, there's always room for more. So this may be a kind of a lazy prediction, but I believe chemists in the future are just going to develop more and more ways to add to this growing library. Another aspect about the future of click chemistry that I want to talk about is the emergence of click-to-release reactions. So as the name implies, this is basically click reactions that have been modified so that when bond formation occurs, some sort of trigger is initiated that causes bond dissociation in another part of the molecule. And in this manner, you've basically built in a molecule releasing mechanism into the click reaction. So I think the most prominent example of this is this modified tetrazine ligation. Just as background, the tetrazine ligation is this inverse demand Diels-Alder cycloaddition between tetrazine and this trans-cyclooctane. And so the modification that's made is that this amine-linked carbamate is basically installed at this allylic position of the trans-cyclooctane. Now, after tetrazine ligation, it's going to form this 1,4-dihydropyridazine, which can easily undergo electron rearrangement to form the pyridazine. But when it does this electron rearrangement, basically the carbon dioxide and the amine becomes released. So as a note, this work was actually developed by this company known as Tagwork Pharmaceuticals, headed by Mark Robillard. And as you can guess, this company is interested in using click chemistry to develop drug delivery systems and also radio imaging applications. So following the discovery of the click-to-release reaction in 2013, we had some really great news in 2020. Basically, for the first time ever, the click chemistry was being done inside of humans for the very first time. So this is only possible because this company here developed their platform known as the click-activated protodrugs against cancer. And how this works is they first have they make this hydrogel, and this hydrogel is functionalized with this tetrazine here. They then take a cancer patient where they know where the tumor is, and they inject that hydrogel locally where the tumor is. Next, they have this trans-cyclooctane doxorubicin prodrug that they administer to the patient via an IV drip. Now, the thing with this drug is that the trans-cyclooctane masks the activity of the doxorubicin, so this prodrug has no biological activity. So it's free to circulate within the human body until it comes in contact with the hydrogel. So when the prodrug comes in contact with the hydrogel, that means the tetrazine is now in the vicinity of the trans-cyclooctane. So the click-to-release can basically occur, which then frees up the cytotoxic doxorubicin moiety. So what you have is basically concentrated drug release, specifically at the tumor. And it's done this way to sort of avoid unwanted systemic exposure to any sort of toxic drugs. So I believe these clinical trials are still ongoing. But at the very least, this gives us a glimpse of a future where we'll be able to do click chemistry within human bodies. And I should also mention that Carolyn Bertuzzi, one of the Nobel Prize winners, acts as an advisor to this company. So with this slide, I'd like to end my presentation. Hopefully, I was able to do justice to the massive contributions of Meldal, Sharpless, and Bertuzzi on the effects that they had on the field of click chemistry and bioorthogonal chemistry. Although I know I didn't cover everything, I hope to at least have given you all sort of a peek into the exciting work within these fields. So thank you for your time, and I'll be more than happy to answer any questions now.
So thank you for the talk. So so you bring up the Lego concept, which is very inspiring. So joining two bricks using some reactions like copper AAC, and then for the cake to release, there's also some reactions. But as you know that just from a naive point of view, two Lego bricks, they can join together easily and then break easily. So I know that the bond formation, why it's so stable, is of course it's thermodynamically stable. So it's not easy to do the reversible reactions. But in anyhow, it is possible that we can combine a cycle of click reactions and click to release reactions to form a cycle chain, so that we can realize really the joining and breaking like Lego bricks. Yeah.
Okay, so from what I understand from the question is if it's possible to, once you click to release, whatever is released then goes and clicks to something else, and so it sort of makes a cascade reaction type of thing. Yeah, I definitely think that's possible, but I don't think that it currently exists in literature at the moment, because it really has to do. I mean, the problem with click reactions is the functional groups are very specific, and that's what also adds to their specificity and sort of the selectivity. So, unfortunately, once the click to release, current click to release reactions are only geared towards releasing amines and hydroxyl groups. And so those functional groups are usually, found in biological systems, so I don't think you can really use them in a selective manner. But definitely in the future, if a click to release can be used to release a tetrazine, for example, or to be used to release isonitrile, I mean, that would definitely be interesting to make a sort of cascading reaction.
Thank you for the talk. Do you have any comments about the stability of azide group in the human body or in any biological systems? Because azides seem to be not very stable compounds, and for how long can they be stable, for example?
That's a very good question. I actually don't know the question. I don't know the answer to that, but I'm sure it should be stable enough because they're doing phase 1 clinical trials. Oh no, that's not with the azide, right? Yeah, probably it's as you pointed out, it's probably eventually going to break down and get metabolized. So maybe that's part of the reason why specifically the click reaction hasn't been taken to clinical trials yet, and it's the tetrazine ligation that has leapfrogged the click reaction in that regards. But yeah, I would have to look up and look at the pharmacological studies on the sort of azide stability.
You said a lot about the contribution of Barry Sharpless in these reactions, and you only said that Morten Mendel he introduced the copper cyclide addition independently from Barry Sharpless, and does Morten Mendel has other contributions? Because Barry Sharpless, as I can see, he contributed in many applications of cyclide addition reaction in many fields, with Fokin, with Finn, and what about Morten Mendel? Do you know which contribution he did also?
For specifically to the click reaction?
Click reactions, only except for the work he published in 2002.
Yeah. So I think with Professor Meldal, his sort of interests were just peptide synthetic methodologies. So he didn't really try to dabble in testing it with biological systems, whereas Sharpless and Bertuzzi sort of went full on into that field. Yeah, I mean, off the top of my head, I can't think of I mean, obviously he used the click chemistry afterwards in order for various combinatorial chemistry type of applications. But in terms of bioorthogonal chemistry, I'm not aware of any types of efforts on his part to test it in biological systems.
Okay, thank you.
But of course, I may be wrong. So, okay. So, is there any other questions? No online questions. Okay. So I guess since we have no more questions, I guess we can end the session. Is that okay? Okay, so great. Thanks, guys, for your attention.